So hi, hello and welcome again. Well, today's topic is the prokaryotic cell. Prokaryotic cells are much simpler and smaller than eukaryotic cells. Bacteria are prokaryotes, and what I'm going to do today again is I'm going to draw a prokaryotic cell and I'm going to label the structures, and then later in the second part of this video I'm going to talk about the binary fission. Binary fission is the cell division of prokaryotes. So when you draw a cell, then I recommend that you always start with the so-called plasma membrane or the cell membrane, because this generally defines the overall size of your drawing. Well, there are some structures here inside the prokaryotic cell that actually go beyond that. Actually, you want to use this color here, and those structures do need a little bit more space. And I'm drawing now the cell in such a way. It is slightly long, elongated. It's a so-called a rod. Rods are longer cells, and but they're different shapes. It doesn't matter. So the structure that I've drawn here, this is known as the plasma membrane or as the cell membrane. So what I'm going to do now is I'm going to simply add a label here, and I'm going to, because I do not want to write it by hand, I've prepared again my labels over here. Some people have problems reading my handwriting. So the plasma membrane surrounds the cytoplasm. And the cytoplasm is the liquid contents of a cell, and there are, it's not only water, but there are, thousands of enzymes and substances in the cytoplasm, and this cytoplasm therefore is very important for metabolism. Remember, all living things have to have a metabolism, so there are many chemical reactions going on inside the cytoplasm. Now, every living thing must have DNA, this cell, prokaryotes also have DNA, but this DNA is not enclosed in a nucleus. Rather, it is a naked DNA that is free floating inside the cytoplasm. Okay, and it's pretty long. The DNA it's much shorter than the DNA of eukaryotes, but I'm just going to concentrate it now here in the center of the cell. And because it is not inside a nucleus, we still need a name for the region where it can be found. So I'm going to make again a labeling line here, and this region where it's found is called the nucleoid. So not nucleus, but nucleoid. But in the nucleoid, that is actually DNA. So, yes, we're not finished yet. There is a second thing that can be found inside the cytoplasm. And these are not made of membranes. The little dots. If you remember what they are, if you've already watched the video of a eukaryotic cell, those dots are so-called ribosomes, and they do protein synthesis. Yes, prokaryotes also must make proteins, and therefore you have ribosomes that are responsible for that. Ooh, this was a really bad. It was a little bit far away. So you know what? I'm just going to erase this here and do it like this here. Okay, so these are the ribosomes. So, what else do we have? Well, we're almost finished, but there are a few more structures that we need. And I'm going to now also draw, where's my pen? Here it is. Outside of the plasma membrane, I'm going to draw a cell wall. Okay. And this cell wall, I don't want to draw it as a line because it kind of looks a little bit then like a membrane. But at the same time, if I start to fill it in like this, you don't really see that there is also a plasma membrane, because it kind of goes, you get the problem. So I'm probably not going to draw it all the way around it. But you get the idea. It has to be all the way around it, because if I go all the way around, then it's going to be a little bit messy. And this here is the cell wall. And the cell wall is made of a very different substance than the cell wall of plant cells. It's actually completely different. The function is the same, that it maintains the shape of the cell. But plant cell walls are made of cellulose, and this one is made of a substance called peptidoglycan, which you don't have to know. Just wants to make it clear that just because structures have the same name does not necessarily mean that they're made of the same material. Yeah. So it goes all the way around, okay? So that is important. And of course, for the labeling, it's a little bit difficult because you have to be able to distinguish between the cell wall and the plasma membrane. Now, I know that some of you might already know that depending on the type of bacteria, there might be also a second plasma membrane around. I get it, okay? We're going to keep it simple here, and we're just going to, tell you the general structure here. Well, okay, some bacteria can move. There are a variety of ways on how bacteria can move, but one way is that they might have protein structures, and either on one end over here, very long protein structures, looks like a tail. It's not called a tail. We call it a flagellum, or plural, flagella. I've written the plural form over here, and therefore I have to draw at least 2 flagella to be correct. Okay. Flagella are, they rotate very quickly, and therefore they're able to push the bacterium forward, the prokaryotic cell forward. Now I'm just going to talk a little bit about some of these flagella here. So you know what, I'm just going to move it over a little bit because I want to make a drawing here. There can be bacteria can have if this is my prokaryote, it can have one flagellum. A bacterium can have multiple flagella. On one side. You know what's even possible? Bacteria can even, or prokaryotes can even have flagella all around the cell. And what happens is they kind of bundle together. They point backwards, and then the bacterium, when it's able to move forwards. Okay, the prokaryote. They're basically moving in this direction. So that's possible. Any combination. You know what? Of course, there are bacteria, prokaryotes around that don't have any flagella. Why not? And nature is very diverse. But I just wanted to make this clear here a little bit. And then is a last structure that is also possibly present. And these are it looks like spikes that kind of go out from the cell. Okay, little spike-like structures. We don't call them spikes. We call them pili. Okay, so where I'm going to label that, I'm going to make one over here, and I'm going to put these are pili, and those pili are also protein structures, and they are responsible for adhering the cell to a surface, for sticking a cell to a surface. So it goes all around it. So how does it look like in reality? So I'm going to move everything up here again to have a little bit more space here. So you have to imagine that a bacterial cell, is not just a two-dimensional cross-section, but it has those pili sticking out on all directions, like a porcupine, okay, in all different directions. You don't see this here because this is only a two-dimensional cross-section. And what do they do? Over those pili, they're able to adhere to a surface. So, for example, if this is a surface, I don't know, the inside of your intestine, maybe. Okay, there is always a danger that bacteria are kind of flushed out, when you go to the toilet. And so some bacteria want to stay in. And what they do is if this is your intestine or any other surface, then those bacteria are able to adhere and stick to the surface, or they are able also to stick together, bacteria sticking together. So those pili are important for adhesion, or cell-to-cell sticking together. Okay, so that's basically pretty much it. What I have here. Sometimes there are exam questions, and then they kind of ask what are some of the similarities and differences between prokaryotic cells and eukaryotic cells. And the most important difference is the presence of a nucleus. So prokaryotic cells do not have a nucleus, they have free-floating DNA. Why is this the most important one? Well, because the presence of a nucleus, or the absence of a nucleus, this is the name-giving criterion. What does this mean? The name-giving criterion. Prokaryote means, pro means before, karyo means nucleus. So it's like before nucleus, it doesn't have a nucleus yet. It won't have one in the future either. But that it's basically the presence or absence of a nucleus is inside the name. And eukaryote, these are the cells of, for example, plant and animal cells, like we have eukaryotic cells. The word eukaryote means true nucleus. So my body cells have a true nucleus, and that is the most important difference. But then again, is some other interesting fact is that we have more prokaryotes growing on our body than we have body cells. Why is that? Well, that is because the prokaryotic cells are so much smaller. Okay, so but I promised you now also that I'm going to talk about binary fission, the cell division that happens in prokaryotes. And I don't have a label now, so we have to write this down. So binary, so here we are, binary fission. Binary fission means splitting into two. That is cell division in prokaryotes. I'm not going to write this down. Cell division in prokaryotes. You do the writing, okay? And how does this work? Well, we have over here a cell, a prokaryotic cell, okay? And what happens is that this cell kind of grows in size now. It grows in length. And not only that, but also the DNA that you can find in it is going to be duplicated. It's going to be copied. So I have to draw now the DNA in here. Okay, and it's a little bit small. And what happens now is that the cell now grows in size. It becomes longer. Maybe I'm exaggerating a little bit here. Because the DNA is now here and is here. Yeah, so the DNA is pulled apart as well as the cell grows. And when this happens, then we have the separation of DNA, but we still only have one cell. So the DNA has to be copied, DNA replication, not cell replication, DNA replication, of course. And then the DNA is pulled apart. And when it's pulled apart, then we have a little space here. And this is now where the cell is then able, you see that it kind of starts to fold inwards here a little bit. Okay. It starts to fold inwards. Yeah, we've got DNA here, we've got the DNA here, and then when we continue this process, then we start to have two cells. Okay, sometimes they separate, sometimes. Now, what's the problem here? You tell me what the problem is. Why am I becoming smaller? This is not correct. There is no reason for the cells to become smaller, so size and proportions do matter. Okay, this is actually one of the reasons why people lose points on an exam question. This is because the proportions are not right. Okay, so and then you have, of course, you have two cells here. Yeah. So what are you going to label? You're going to label, of course, inside here. This is the DNA. Here you can say that the cell grows in size, and here this is when the cytoplasm starts to split. Yeah. So in that sense, binary fission and eukaryotic cell division called mitosis have certain similarities because the cell's DNA has to copy first, it has to be separated, and then the cell, of course, also has to divide physically. Yeah. So this is binary fission. This is cell division in prokaryotes. Because it's so important, maybe I'm just going to write it anyway. Okay. It's cell division in prokaryotes. Okay. I have to be honest, I didn't quite like the drawing that I made, so I prepared a little bit some cutout pieces here. So this is supposed to be my prokaryotic cell. In here we have the DNA, and as the cell grows in length, also the DNA is copied. Look what happens, the DNA is copied now, and now we still only have one cell, but there are two pieces of DNA, two DNA molecules in the cell. And the cell continues to grow, and see how it starts to fold in here? On the side, yeah. So you see the cell membrane is pulled inwards. A new cell wall is being built and constructed as well, yeah. And we're almost finished. And see, now we have two cells, okay. And those two cells either stay together or, in many cases, they completely separate, and then the whole process is able to repeat again. Yeah. So this was basically it. I hope that you again liked the video, and see you around in the next video. All the best. Bye bye.